Thank you so much, King Fai, for your very kind invitation and a very embarrassing introduction. Thank you very much. It's a great honor for me to visit U of Michigan here in Ann Arbor. This is my first time in Ann Arbor. But the first thing I want to tell you is why I consider this as a distinguished honor, because I was supposed to be here 17 years ago. Okay, so it's a visit that's delayed by 17 years. So 17 years ago, I was a postdoc fellow, looking for jobs, right? Dr. Omenn called me up and gave me an interview opportunity. Everything's scheduled, and I got sick, okay? I got a high fever, so I couldn't make the trip. I was trying to reschedule it, but the timing didn't quite work out, right? I have to take an offer from this place, right? Didn't get to visit Ann Arbor until now, right? I mean, this past 17 years passed, right? So I made lots of friends, actually, with Michigan people already, including one of my own mentor, John Moran, right? Yeah, I mean, I learned pretty much, I started studying transposable element by accident, okay? And John was the person who led me into the field. I don't think, John, you remember, I called you up when I started my lab. You read my first R1. We had so many conversations at different conferences. So thank you so much, John. So this is a distinguished honor for me to be here to see the place that I was supposed to visit 17 years ago, and to see mentors and lots of friends that I made over the years. As King Fai is also a friend. King Fai mentioned I work quite a bit in consortium. So I'm going to talk a teeny tiny bit about this consortium work, but really trying to bring a kind of my philosophies towards team science, right? And I think every single consortium I work for, right, I have a collaborator here in Michigan, so this is really, again, a special honor. Right? So I ended up, going to St. Louis and start my lab. Anyone been to St. Louis before? Right, we're not too far away, so there's still like a couple of direct flights between Ann Arbor and St. Louis. And St. Louis is very well known for this gateway arch, right? So this is the kind of the gateway towards the Wild Wild West. So in fact, this is a monument recognizing Lewis and Clark walking all the way from St. Louis to the West Coast, right? Pacific West Coast for the discovery. So we use this as a symbol for one of the projects that we have called WashU Epigenome Browser. So that was a browser we developed for the roadmap project. It's still going strong, very innovative. An important part is we host a ton of human epigenomics data, hundreds of thousands data, right? So, I invite you to take a look at the browser, maybe the data there and the function there might be useful for your own research. If you found a bug, or if you need a feature, you can talk to me directly, Slack me, email me, because we're the developers, right? So we're very excited about the browser. I'm not going to talk about the browser. I'm mostly, sharing with you kind of the nerdy part of myself, studying transposable element, right? So this particular title actually came from a recent review article called Unmasked, Transposable Elements as Drivers and Targets in Cancer. So I stole that title from my student who wrote that review. All right, so let me get started. So where do I start? I usually start this with the human genome, right? So this is the human genome. If you print that into book volumes, this is how many books it takes to write the blueprint of humanity, right? So this is also the product of the Human Genome Project, right? I can't emphasize enough. I do not need to emphasize how important this project is, right? First of all, it is a team science, okay? It took a village to put together this resource for the community, right? That's the value of team science. Second, this project really transformed how we do biomedical and biological sciences, right? And WashU genetics played an important role in sequencing the human genome. We sequenced about a third of the genome. So I feel like very privileged to be in that environment to carry that legacy. For me, when I was a kid, I loved reading books, right? So it's probably not a surprise. Now I'm trying to read this book of genome, and I often, make an analogy using the different ways of reading books to describe epigenetics, which is what I mostly focus on, the epigenetic regulation of transposable element. Before I get into transposable element, I want to use this opportunity to give folks a quick update of where is Human Genome Project, right? So we all know the impact of this project, right? So from 20 plus years ago. But this project, like any other project, has limitations, right? So in fact, one of the first things that students raise their hand in classroom when we talk about Human Genome Project, they ask, hey, whose genome is it? Okay, whose genome is it, right? So it's, some people say, oh, it's James Watson's genome, Craig Venter's. No, right? So none of their genomes, right? So it's actually nobody's genome, and that's actually a limitation, right? Turns out, most of the sequences come from, depending on how you count, 6 to 20 individuals, right? So mostly from 2 people, right? But if you count everything, there's 20 people's genetic information in this genome. And then you ask, who are these people, right? Why their genetic information makes up this blueprint of humanity, if you will. So it turns out the only thing that's common among them is that they all lived in Buffalo, New York, back when Human Genome Project was launched, right? So in fact, this is the advertisement in the local newspaper. They looked for 20 volunteers, they got their blood made into cell lines, and from the cell lines, you isolate DNA and then make single bags and yaks and then sequence them, tile them, polish them, and boom, you got your human genome, okay? The reference human genome that everybody uses today, okay? So as a result, it has a few limitations. Number one, it's not even a full genome, it's a half of genome, right? So we have like 2 haplotypes from mom and dad, 2 sets of chromosomes. This is a single haplotype. It's mostly a mosaic, right? So coming from different individuals. Therefore, from a reference point of view, it does not represent the genetic diversity that we have. It's not complete. It misses about 8 to 10% of sequences. If you look at centromeres and telomeres, they're mostly ends. So that's sort of being completed by this recent T2T consortium to complete the human genome, but that's still a single haplotype, right? And another important limitation that I care about quite a bit, it's not like broadly talked about, is that there isn't a single cell on the planet that has this genome in it, but we kind of make an assumption, right? Whenever we have a sample, we have we do our bioinformatics, we make an assumption that the sample we have has this genome in it, okay? But that's not true. That's an important assumption for us to move forward, but it's a limitation, right? So I think I want to use this opportunity to give folks a quick update of how we're dealing with this problem, and that is one of the consortium projects that I'm very, very excited about, the Human Pan Genome Project, right? So the project actually started 2019. So the first phase was completed as a success. So we just started our second phase. So it is a large collaborative work, including players from US as well as internationally. This is Washington right here, so we look pretty good on the map, right? So right in the middle of the continental US, kind of far away from mountains and oceans, but that's okay. So what do we do, right? So we sequence individuals' genome, okay? So the goal of the project, the first phase is to sequence complete T2T genome of 350 individuals. Okay, so that's 700 haplotypes, right? So that's the data production, right? We're not going to throw 700 genome references to King5. We're gonna fold that into a data structure that's this mathematically defined graph, right? So like the bubbles represent variation, something like that. So so that's sort of the other component of the project, right? So we did pretty well. This is a review article we published at the beginning of the project. First year of project, we basically did technology benchmarking, right? So tried, kitchen sink, right? Try everything, and long-read sequencing, right? Really came into the center stage. The product from that is the first deployed human reference genome. A celebrated moment was now 3 years ago when we put out the first draft human pan genome references. So this one contains 47 individuals, right? So 94 haplotypes. So I think this is already gaining quite a bit of traction. So the last bit I want to talk about is where we're going, right? So we completed phase 1. We're now at the beginning of phase 2. This year, we're putting out the second release. In fact, all the data is already publicly available. So we have 470 haplotypes at this point, right? So from a data production point of view, I think we figured out that all the kind of a problem, so we can just go forward. So we're pushing towards 1,000 haplotypes. But the other important component, there's 2 more components that are taking center stage in the second phase. Number 1 is the bioinformatics, right? So think about, how do you deal with now, hundreds of reference genomes. So essentially, most of the bioinformatics tools that we've been using need to be rewritten, right? So if you are a bioinformatician, good news, you have job security. But the other thing is, going back to the nature of this project, if we're building a resource for humankind, it cannot just be a US a project. It has to be an international project, right? So this is not the best time to push for international collaborations, but I actually putting a ton of effort to try to grow and then build an alliance across the globe to work on this project together, right? So I call out to partners to really join force to build this resource, even beyond the lifetime of NIH-funded human pangenome reference. That's all I'm going to say about the pangenome. I, again, invite you to check it out. I think it's going to make a pretty big impact on genomics research, right? I have many anecdotes that I can share. So I spend a lot of time doing this, building these resources, right? And another resource that I think many people know about, and I also contributed early time in my career. In fact, this project contributed to more of my own career development than my contribution to the project. That is the roadmap project, right? So this is one of the post-genome project where we are building the human reference epigenome, right? So started as this US epigenome roadmap project. This is the transition time for me going from postdoc to independent investigator. We developed the WashU epigenome browser to host this data. So the project was considered a pretty good success. It went from 2008 to 2015. So the roadmap project ended, and then we generated, reference genomes for representing hundreds of cell types and multiple individuals, tissue types, developmental stages. But we also formed this international human epigenome consortium. So the consortium continues to go and absorbing new memberships, including Epi Hong Kong, ENCODE project, 4D Nucleome project. So even the roadmap project ended, right? So the consortium continued. So this is why we have hundreds of thousands of human epigenomic data sets now accumulated for people to use as a benchmark, to do more analysis. That's, most of these are being hosted at WashU epigenome browser. So I think that the message I kind of want to send is, we're all scientists, right? But we also have a responsibility to build a community, and this is how we build this community, and we benefit from this community, right? How did I benefit from this, right? So the first thing I benefited from was all the great data that's freely available to me to look at transposable elements, right? So many of you probably remember the early days, when we have sequencing data, you map to the genome and then you throw away rates derived from repeats, right? In fact, one of the first surprises from the Human Genome Project was, the coding sequences is just 1.5% of the genome. Majority of the genome are non-coding. At least half of them are coming from transposable elements, science lines, and LTRs. In fact, some people argue that a lot of the sequences over here are derived from transposable elements as well. It's just they're too old for us to recognize their evolutionary origin, right? In the context of epigenome, if you just think about CpG sites, right? More than half of the CpG sites are, they're from transposable element. In fact, some people argue that the primary role of DNA methylation is to repress transposons, right? So everything else is collateral, right? So genomics is moving rapidly, but our understanding towards transposable elements are kind of, limited, but also for good reasons, right? So early days technology-driven genomics started with microarrays, but probes really, do not represent this repetitive region of the genome. So even if you hear, hey, we're doing a whole genome tiling array, for human, that's really just half of the genome, right? Lots of the repetitive region was not being probed. Second generation sequencing clearly took over the field. You read this thing probably 1,000 times, right? 60% rates mapped to a unique region and rest are discarded, right? So we discard them because, we don't know what to do with them, but also because, conceptually we feel that they may not be that useful. When I was a PhD student, I was trained a computational biologist and I did motif finding, right? I wrote computer programs, 20,000 lines of code. I actually wrote them, computer programs to predict transcription factor binding sites, right? And the first thing you do before you do motif finding is to do repeat mask, right? Because they're gonna overwhelm, your statistics. So this is the ironic part, right? So my entire career is now built on the stuff I throw away as a PhD student. Okay, so for you never know where life leads you, right? For anything. So we throw them away because they're difficult to handle, but also because people call them junk DNA. But are they really junk, right? So this is Barbara McClintock. She won the Nobel Prize for the discovery of transposable elements. She started this field, right? But when what did Barbara call them? Right? She called them controlling element because they alter gene expression. That concept, I think John agrees with me, deserve another Nobel Prize, right? So that concept did win a Nobel Prize. A small piece of DNA can change gene expression. Unfortunately, it didn't go to Barbara. This is Eric Davidson, Roy Britton actually proposed this evolutionary theory that why we have repeats. There's no experimental data, right? Why we have repeats, right? So they coordinate the gene regulation, right? So the repetitive nature provides that, right? So I really spent, the past 15, 20 years really thinking about this. I think the field really now, I think through the power of genomics, of course, there's a ton of, noise in that as well. But I think we all kind of come to this realization that, transposable element might be called dark matter or dark energy, very similar to the way that the material world works, right? So they are the dark matter, dark energy in our genome. They're full of energies, right? So early days we really got started looking into the type of innovation because they're new stuff, right? New material that are bringing in, right? So they provide the genome a lot of opportunities to innovate. Okay, creating new genes, new proteins, but more broadly is to provide this regulatory innovation. So we spend a lot of time entertaining that idea. And then we also come across how it's really, transposable element depositing like transcription factor binding site, providing that kind of opportunity, providing new regulatory relationships. But then we also come across how they mediate turnover events to the degree that they may provide a mechanism to maintain genetic system, to provide this robustness, right? What I'm going to present today mostly has something to do with how they might provide the material, how to allow epigenome to evolve, right? So it's very vague, but I'm going to use cancer as an example to illustrate some of our thoughts around how epigenome evolve, right? We kind of know how genome evolved, but what exactly do I mean by epigenome evolution? I'll get to that with some simple illustrations. Okay, so that's what I'm going to mostly present around transposable element. This is the opportunity for me to go back to the memory lane and tell you that accidental finding that got me started. So when I was a postdoc, really by accident, I found that, in human, p53, I was studying p53. p53 doesn't need the introduction. It's such an important molecule, right? So guardian of the genome, tumor suppressor gene, 50% of human cancer have mutated p53. That's how important p53 is. And it's a transcription factor. It binds to a motif. And that motif, right, that binding, 30% of p53's genomic binding site in the human genome were evolutionarily introduced by a virus, right? So this is a endogenous this is a virus entered our ancestors' genome and become an endogenous retrovirus. The virus was like, jumping around. This is a transposon, right? So it jumped, there's probably reinfection as well for about 20, 25 million years. We developed a lot of epigenetic mechanisms to shut them off. So about 25 million years ago, this transposable element died, right? So there's no more activity from that point on. So this 20 million years of jumping, right, left behind 30% of p53's genomic binding site, right? So that accidental discovery really changed how I think about gene regulatory network evolve, right? I'm not saying these sites are functional. There are a few sites that got validated to be functional, but it provides that opportunity. So that's a mode of evolution for, in the context of gene regulation, right? That's sort of this beginning of a whole, my investigation toward, into the relationship between transposable element and gene regulation. I'm happy to say that till today, we're still studying p53, right? So this is actually one of the most recent publications from the lab where we're characterizing the relationship between p53 and transposable element expression in the context of cancer. So of course, we moved beyond p53, right? So part of the effort really goes to trying to see if this is generalizable, right? So you would predict that this is not unique to p53. You would predict this is not unique to human. So we started looking into other systems. And the interesting part is, it doesn't matter where you look at, human, mouse, rat, all the way to zebrafish. They have very different transposable element, right? But if you ask, do you see these transposable elements behave like regulatory elements? And the answer is always yes, right? So to the point where the most recent ENCODE data, if you probe that data, you'll see that 20 to 25% of regulatory elements are derived from transposable element, right? So this is not just us, plenty of people in the field start to make similar discoveries, and that concept, that observation start to solidify, right? To the point where we feel that genomics has really given us a lot of material to work on, and what we really found is going back to the ability of transposable element to introduce genetic material, right? And this material can be used in many, many different ways. They're not designed for that purpose, but it happens, during the course of evolution. So these are I start to, write. These are a few, review articles that I got involved in writing the past few years, right? One of them really says, doesn't matter which function that we look at, right? So, enhancer, promoter, insulator, and whatnot, you're going to see transposable elements involvement, okay? And then the article on the right really focused on the role of transposable element in shaping the three-dimensional genome structure. It's really mediated by its ability to provide binding sites as anchors for the 3D genome. So we start to, really, excited about now transposable elements are no longer being ignored. Lots of people are beginning to show interest in this, and they are an integral part of this functional genome, especially in the context of evolution. So most of these are done in the context of evolution. So that's great, right? So I love evolution. I'm an evolutionary biologist by heart. But we're in the medical school. We think about human diseases all the time. So we start to think about whether or not transposable elements are important, not just for evolution, but for human disease. By the way, anyone recognize this cat? All right, this is, I made this like a little bit over a year ago. This is Taylor Swift's cat. This is the cat that I was thinking that may change the political landscape, the childless cat lady thing, right? So it didn't, but then, I haven't got around to change it. I did change that, this meme, you'll see in a few minutes, but a little joke here. Anyway, so we decided to, think about, so we've been thinking about transposable elements' role in the context of the evolutionary a lot, right? So naturally, we start to think about, disease as a process of evolution, right? So then this is how we got into cancer. But before I'm showing you the cancer work, I want to show you, really the logic that got us into this. That's epigenetics regulation, right? So look at these two animals. So this is the most well-known example when you talk about epigenetics. These are agouti mouse, okay? I mean, they're lovely, right? So it's, especially the one on the left, chubby, golden fur, you almost want to snuggle them. So you talk about epigenetics, you will come across with these examples. I think the commercial value of these animals were only recently realized. It was this La Poo, right? So look at this too, right? So anyways, these two lovely animals, right? Why they're famous? Because phenotypically, they're clearly very different, right? Unfortunately, this one on the left has a lot of issues, right? So high risk of cancer, obesity, and all that, right? Genetically, they are identical, right? So every base in their genome is the same, right? So, if we believe genetics, why they have same genome, different phenotype, right? So, turns out the only difference between them is their mother's diet, right? So, the mother of this animal, just eat the normal mouse chow, but this one had his mother had like vitamin supplements, right? Especially B12, folic acid, providing as a meso donor. So as a result, this one is more wild type looking. So this is a good example of saying like you're not who you are, you're what your mother eats, right? It's also give us like really good excuses to blame our parents for the stuff that we can do, right? So I teach this all the time when my kids were little. They come to classroom when there's just like a school's out, so they learn this and they use this against me now. Anyway, so what's the molecular mechanism behind it, right? So is that this is the causal gene. Causal gene is agouti. The genetics, the mechanism behind this is a transposable element. Okay, so this is the causal gene agouti. About 100 kb upstream of agouti, there is a transposable element insertion called IAP element, right? So this element can serve as a cryptic promoter. So transcription can start from here and go all the way across this 100 kb and then resulting agouti gene overexpression, right? So that's the causal. So when agouti is expressed at the time and location where it should not be expressed, that tied to all the bad phenotypes that we talked about, right? So when the mother takes, say, folic acid as a supplement, it provides some meso donor. So the baby will be able to methylate this IAP element, right? So once this element is methylated, it cannot serve as a cryptic promoter anymore. So agouti expression returns to normal, you get a normal phenotype, right? So the phenotypic difference is not due to genotypic difference, right? Genetically, they're identical, right? But rather they're due to methylation differences, right? So this is something on top of the genome. That's why we call it epigenetics, right? So it's a beautiful example in epigenetics. I think lots of people, everyone in epigenetics knows about it, but I think not everyone in the epigenetics field realize that this happens on the transposable element, right? And 50% of the genomes are made of sequences like this. So you can imagine, speculate, how often, this mechanism may be at play for diseases, right? So lots of diseases we don't know, what's the causal, what causes them. Every gene, there'll be some transposable element around. So you can sort of imagine this. So this is actually how we started. We decided to, hunt for the human agouti, right? So we want to look for a human disease where, the causal factor is not genetics, right? It's epigenetics in nature, right? It's some epigenetic changes lead to transposable element activity differences that leads to gene expression differences, and that causes disease. So where did we start? We started with cancer, right, for a few reasons. Number one, there are just a ton of publicly available data out there, right? TCGA is another consortium that generated all this data for the community. Number two, we know there are some hallmarks of cancer. For example, every single cancer will need to activate oncogene, right? So the mechanism of upregulating oncogene is studied for 30 years, written in textbooks, right? That's mutation, translocation, and amplification. So what we hope to identify is the examples, where oncogene activation is not immediately due to a genetic event, but rather due to an epigenetic event. So when we started this, there's actually a few examples in the field, right? But less than 10 examples where people have shown that a transposable element become demethylated and start to express and get into nearby oncogenes and result in the upregulation of that oncogene. So that's what we set out to define, but in a more kind of a comprehensive pan-genome manner, right? So we're really looking for how common is this type of mechanism. In other words, oncogene is highly expressed, but it's really coming from a epigenetic event from a nearby transposable element, right? So you can think about this as looking for that agouti gene, where the high expression is the causal for disease, and then the expression really comes from a transposable element. So to do that, we took advantage of TCGA data. Like many people look into TCGA data, we just look at it a little bit differently. Instead of just mapping them to gene structures, we took a deep tour. We did transcript assembly. This allows us to see the alternative transcript to get to the 5' end of the sequence. So and it turned into a series of tools now we call TE promoter finder 1 and 2 and 3. So these are 3 generations now. The most recent generation works on long read, works on single cell. But the concept is the same. It's like, look for alternative transcription start site. So we're not surprised that we're going to find some, right? But what we were really surprised with is, how broadly this phenomenon is, right? So 50% of the tumor we looked had at least one oncogene where we found the high expression of that oncogene is really coming from an alternative promoter that's provided by a transposable element. We believe this is an underestimation because we rely on typical RNA-seq data, which is oftentimes 3' biased, right? So these are some examples. So I'm just going to use one of the examples to illustrate this phenomenon, right? So I'm going to look at the last one on this list, number 10 on the list, called ALU-JB Lin20B. Okay, so Lin20B is a bona fide oncogene. Okay, so it's actually highly expressed in many liver cancer and some lung cancer. It's pretty broad. No mutations ever reported for Lin20AB, right? So what we found is this highly expressed Lin20AB actually start from a transposable element called AlloJB about 20 kb upstream, right? So transcripts start from here and go all the way into the actual canonical gene, and that's really the reason that Lin20AB has been is highly expressed in many of these cancer types. Okay. So what are the values of these findings? Immediately, they have some prognostic values already, right? So again, using the Lin20B example here in liver cancer, again, this is the actual Lin20B transcription coming from AlloJB going into Lin20AB. So patients with canonical Lin20AB feature much better than patients with this AlloJB Lin20AB, okay? It's actually not difficult to explain, right? Lin20AB is an oncogene. This version is highly expressed, right? So having more oncogene is probably what makes this tumor more aggressive, right? So at least at this point, there's already some kind of a prognostic value. We believe this is an epigenetic event, so we went in and then profiled a bunch of epigenomes. So in the end, we actually did like long-read RNA sequencing to confirm this particular isoform. So over here, this is the canonical Lin20AB promoter. Over here is the actual promoter that tumors use to express Lin20AB, and they're lowly methylated in these representative tumor cell lines. If you look into other epigenetic signatures, and this region actually, just over here, this region is, has all the canonical classic promoter marks, including K27 acetylation, K9 acetylation, open chromatin, and transcription start site signal over these transposable elements. If you look at normal tissue, again, this is where the roadmap project played an important role. So we need to see what happens in the normal tissue, right? So what you're looking at here is like demethylation tract. These are whole genome bisulfate data. Ticks represent CpGs, and blue color represent methylation level. So you're going to see that this whole region, as expected, these are transposon. In normal tissue, they're just methylated, right? So presumably repressed, but in tumor, they become demethylated. Right, so epigenetically, we see that this transposable element region is gaining this promoter signature. So what I haven't told you is this two transcripts, right? So these are two different transcripts, right? So even it gives you the same gene, this Lin20AB protein, but with a small difference, right? So this tumor-specific transcript coming from AlloJB actually skipped the first exon. Okay, and go into the second exon. So as a result, the protein misses 3 amino acids on the N-terminus and gained 22 amino acids coming from this AlloJB sequences. Okay, we do not believe there's any functional impact, right? So we did some, but the evidence we have is later on we did some rescue assay to show these two proteins are equivalent. But the size of the protein is a little bit, right, different now, right? So you can run a plain old Western blot and be able to tell them apart, right? So on the right here, these are the canonical Lin20AB protein. And over here, this is the monster protein that we found in cancer. Now, these are the cell lines where people have been using for a decade to study Lin20AB and publishing on Lin20AB, right? Without realizing that they're actually studying a slightly different protein, okay, which is this one, right? So this little difference gives us make our life a little bit easier to do additional validation experiments. So this is where we take advantage of, again, technology development. This is CRISPR. Everybody and their cousin are CRISPR things out from the genome. A few of us CRISPR out junk DNA. Right, so this is the case where if we take out promoters of Lin20AB, okay, nothing happens, okay, because it's actually not used as a promoter anymore. But if we take out AlloJB, this transposable element, this junk DNA, protein's gone, right, because that's actually the real promoter, right? So to the degree where if we take the resulting cell, if we take out Lin28B promoter, the cells still form tumor in a xenograft model. But if we take out Lin28B, these cells lose their ability to form tumor, right? So this illustrates that these allo-JBs are kind of the genetic factor behind, this tumorigenic ability of at least these cells. Okay. So what about epigenetics? I've been saying about this is epigenetic regulated. So we want to go ahead and then try to prove this, right? So so the experiment we did were the following, right? So the first, we did site-specific methylation, right? So the idea is you take a Cas9, that Cas9, you hook it up to a methyltransferase, and you can methylate whatever you want in the genome. Okay, so we try to methylate this allo-JB element. This turned out to be a pretty difficult experiment. If you think about it, we're selecting against ourselves, right? We're shutting down allo-JB so that we're shutting down Lin28B, which is pro-growth, right? But we want to screen for live cells, so we're going against the stream. So it's a difficult experiment. We repeated it multiple rounds, and the best we could reach is to increase the methylation by 25–30%, and we see correspondingly a 40% reduction of expression, right? So we found that it's maybe good enough, right, to illustrate the point. So we have to do the other way around. So we took a cell line where the allo-JB is actually methylated, right? We did a site-directed demethylation. Okay, that experiment worked out beautifully, like a charm, right? So within a few days, right, we see the methylation level drop to 0. Okay, and not only that, the cell lines start to express this slightly bigger, this lot bigger protein, this monster protein, right? So that's great, right? So first try it worked, and we thought CRISPR has higher efficiency in the cell type. We should always use this cell type, right? So that's the first, immediate thought. And if you think a little bit more, you realize that this simple experiment is not telling you about the CRISPR efficiency at all, right? It has nothing to do with efficiency. This actually tells you how tumors evolve. Okay. Now we all know cancers, they're a process of evolution, right? You've seen the clonal expansion trees many, many times. Every single tree out there in the literature is drawn based on genetic data, clonal expansion, okay? But cancers evolve epigenetically, right? You don't need every single cell to lose methylation. All you need is a few cells few alleles lose that methylation, because that's going to result in overexpression of Lin28B. And the daughter cells, will gain the ability to progress a little bit faster. So that's going to be, behind this clonal expansion, but on an epigenetic level. So I think this experiment actually taught us how cancers evolve epigenetically, right? So there, part of this lab is our lab is really we hope to be able to draw that epigenetic evolutionary tree. We don't know how to do that yet, but this is really telling us there is a parallel tree, to the genetic clonal expansion tree. Okay. So now we're back to evolution. I just told you, like, evolution is cool, but they don't bring cash. So we have to think about, how does this knowledge benefit patients? And there are really exciting opportunities. So first, right, the one event I've been talking about so far is a transposon transcription going into nearby gene, which is an oncogene, and form this chimeric transcript. Now, this transcript is tumor specific, okay? This actually gives us an opportunity to target it, right? So targeting oncogene is always a dream, but it's very difficult. Part of the reason is, oncogenes are also normal genes. They often play an important role in normal cells, right? So how do you specifically target the version that's in cancers, okay? So this is Noah is an MD PhD student. So this is what he thinks about more often. He decided to try using splice leaping switching oligo to target these transposable element sequences to bring down this oncogene transcript level, right? So this is preliminary data showing that, this is positive control for killing cells. This is how tumor cells grow, right? If you use a antisense oligo against this allo-JB lin20B, you can effectively inhibit this tumor growth. But if you use scrambled control, the tumor will continue to grow, right? So this is, at least in this setting, it's effective. This is dependent on allo-JB lin20B, because if you take a different cell line without allo-JB lin20B, you don't see this effect, right? So this is sort of some preliminary data to show this is working. And then he moved on to do an animal model. Again, the preliminary result was pretty exciting. This antisense oligo is able to slow down tumor growth and extend the life of these animals, right? So, he's actually really eager to, put this in a patient and say, no, let's repeat this experiment a few more times. So this is exciting, right? So this is a potential method or therapeutic method that's orthogonal to anything that we've had now, right? So we all know, to kill, cancer, so it's no longer like one target at a time. You would never kill cancer that way. It's really having multiple things happening at the same time. So at least this sort of provide an alternative way to target tumors specifically, right? The other kind of exciting part is this is just one candidate, right? We know in different type of cancers, there are quite a few oncogenes that are expressed this way. So maybe there is a broad way of kind of targeting that. Okay, so that's pretty cool, right? So, but this is actually not the coolest, right? So what's actually more exciting about this is going back to the same event where you have a chimeric transcript that's cancer specific, partially transposon, partially gene, right? So when this is oncogene, it's back, but oftentimes this is not an oncogene. This just happens in tumor cells. This chimeric transcript sometimes gets translated, right? And that's going to sometimes result in a chimeric peptide that is cancer specific. So what is this? This could potentially be new antigens, right? So new antigens is a big deal, right? I think people are now realizing the power of immunotherapy, why we sequence all this cancer genome to get the mutations that give you that new antigen so you can, treat patients with CAR T or other immunoassays. So that works like a charm for cancers with high mutation load, like melanoma or lung cancer. But there are plenty of cancer with low mutation load, like pediatric cancer, glioblastoma, right? So oftentimes you don't even get to that mutation that gives you that new antigen, right? This is why I think lots of people start to go after splicing, go after other dark proteome, right? To look for alternative antigens. Now, this is an alternative way to generate new antigen, right? So it's the same event, but now we're looking at this chimeric region, which could potentially be a source of new antigen. So we kind of started another project, a hunt for this new antigen, and I think I need to actually hurry up. So the idea is very similar, right? So it's to look for this potential new antigens derived from a chimeric transcript. Short story, we found a lot, okay? And we also have to be very careful to make sure we have the right controls so they're not in normal cells or during developmental stages. Even that, we ended up with like, 1,000 candidates. I'm gonna skip some kind of analysis, but to say, what's the most exciting part of this, right? These antigens, new antigens, predicted ones, they're much more recurrent than mutation-driven new antigens, right? So mutation-driven new antigens are often private, okay? That's why we do like personalized medicine, right? So across TCGA data, TCGA sequenced 10,000 tumor, right? The most shared mutation-driven predicted new antigen is shared by 82 tumors. Here, we have more than 200 candidates that are shared by more than 200 tumors, right? Some candidates are shared by like 1,500 of the 10,000 tumors. So they're more recurrent, which gives us a kind of a direction where we can perhaps, develop vaccine strategies, develop, on-the-shelf therapeutics to treat patients, right? So these are some of the validation experiments. So this is actually a candidate that we showed to be presented on the surface, and now we actually have both BCR and TCR and T cells that can kill this cancer cell. So this is in collaboration with Daniel Di Carvalho in Canada, right? So this is sort of giving us this very kind of a exciting direction, thinking about on-the-shelf vaccination for cancer, right? So that's very cool, right? I would say that's still not the coolest. So I want to bring the sort of the final bit of excitement, right? So new antigen from mutation, that's a genetic event. When you don't have new antigen, can you create more new antigen by creating more mutations? That sounds dangerous, right? But guess what? People are doing that, right? Patients are giving chemotherapies for the purpose of creating more mutations to turn a cold tumor hot, okay? Because we're desperate, right? So it's dangerous, but we're doing this. But this particular mechanism, as I mentioned, is epigenetic in nature, right? Epigenetic events are slightly easier to control than genetic events, and they're reversible. So this gives us a direction where we can use epigenetic therapy to trigger production of particular new antigen, right? When the patients, the idea is really, when a patient comes in, right, you profile them, right, and you have enough knowledge base to say, all right, you have this particular target, so we have something on the shelf, so we can treat you. Or even if the patient doesn't have that target, right, we have enough to predict that we're going to give you this particular combination of drugs, so that a week later, right, your tumor cells will start to express this new antigen on demand, on target, right, where your normal cells are okay, right? So that's what we define as precision medicine, right? So can we do this, right? So in fact, quite a few years ago, we already know that epigenetic therapy can target transposable element. Actually, they prefer to target a transposable element to kind of reactivate them. This experiment, I already showed this experiment for a different purpose. This actually shows that we can specifically activate activate a chimeric protein. Okay, this is Lin28B, so we're not gonna use this, but this as a proof of principle, we can target and activate specific new antigens, right? So, we kind of did a preliminary study using glioblastoma stem cell as a model. This is when the technology is moving towards long read. So this part will just be long read, and the result is standard epigenetic therapy increase candidate new antigen that we can target by 3 to 5-fold, right? And using CRISPR-based method, we can target them, right? So CRISPR delivery is another problem that I'm not dealing with, but the field, I hope there's plenty of people, going after that, right? So, I really just to bring this back, I think, the past few years, we came from studying the evolution of transposable element in the context gene regulation, but we come across, if you use the same framework to look at cancer, you realize that, cancers are evolving fast, right? So they take advantage of this transposable element to do stuff that's beneficial potentially for them, right? So this is why this genomic dark matter, there's really double-edged sword, right? They're providing opportunities for the tumor cells, but they also provide opportunities for us to target them. So this is a kind of a new frontier that got us really excited, right? So coming back for a quick summary, I'm excited about transposable element. I will think they're full of energy. We had lots of fun, looking at the regulatory innovation, impact on genetic robustness, and epigenome evolution. I'm using just cancer as an example to illustrate the kind of epigenetic changes that driving phenotype and transposable element is actually providing that plasticity. So these transposon-derived antigens are a result of reactivated T promoters. They're more recurrent and they are epigenetically targetable. I think I'm running out of time. I actually have a few more slides talking about other consortium project. I'll leave this to I think the other one other benefit of this, I start to recognize the value of bringing team sciences and many teams together, right? So these projects that Ting Wang introduced, they all started independently, right? Again, I have collaborators, friends from U of Michigan in every single consortium, right? And now we found that by integrating them together, for example, the human pan genome are already providing new references for IGVF, for SMART, and for MOHD, for these different consortiums. So already increasing the value of their data. And if there's another consortium I want to work for before I retire, that would be the human pan epigenome project that I'll try to lobby over the next 5-10 years. I think I'm running out over time already, so I'll give the acknowledgement credit where it's due. I'm very fortunate to have a team of young investigators who are brave enough to really do things that they do whatever they want. They don't even talk to me about them. Like I said, I work quite a bit in the context of different consortium. I didn't actually introduce all of them. These are some of my collaborators on in cancer funding, and finally, I always want to propose. I've been proposing this for a number of years, to put Bioinformatics McClintock's image on a U.S. currency. With that, I want to thank you for listening. I apologize for running a little bit over. Thank you very much.
Thank you so much for this wonderful talk. Question time?
Better raise. John's gonna give me questions.
Ting, thank you. That was a fantastic talk, very innovative. I guess the question I have is that most of the places where you're making transcripts that could generate putative neoantigens are from old transposable elements in the genome. So if you go back to your epi maps, do you ever see these alternative transcripts made in, say, stem cells, which could be the origins of some cancers? And if so, I know they're new, but are they non-self? So are these new transcripts, if they're expressed in other tissues, going to truly be recognized as non-self to qualify as a target for the therapy? So do you have any thoughts on that? I know that's like $1 million question, right?
Yeah. Fantastic question. This is the question that keeps me awake at night, right? So short answer is we don't have a complete picture of this. We're doing our best to look for as many as possible, right? So we implement very strict filter, right? So anything that we see anywhere during normal development in a obscure cell type, we're deprioritizing them, right? That said, testis, that's okay. So there are a few tissue types where we don't use them as strict filter, right? So two things. One is, yes, I mean, we do see waves of transposable element transcription activities in early development, in a certain specific cell type, right? So in the context of cancer, applications in cancer, we filter them out. Okay, we filtered them out, right? So we're excited about them. We're trying to understand why they happen. What are the epigenetic environment, what are the sequence variation that make one copy express, not the other. So we're doing a whole lot of that, right? But in the context of developing a therapeutics or vaccination, right? We simply filter them out. Okay, yeah, sure.
Thank you. Okay, we have another question.
Thank you for the fantastic talk. I wonder whether you can comment on potential resistance to therapeutics derived from transposable elements, like whether it's like in the future, like if tumor may develop some resistance, what may be happening, and it's like whether there's any like additional remedies.
Also a great question, right? So I have also two layers of answers to this, right? So the first one, the simple one, is I think the kind of resistances cancers will develop is not going to be different from other type of things you go after. They evolve very fast, they adapt. So this is why I feel that it's not like creating one new strategy, it's rather to have like multiple angle to target cancer. Okay, so that's one easy and short answer. But I think this does open up an entirely different thinking process I don't have time to go into, right? So, tumor cells, they take advantage of these transposable elements to do what's beneficial for them. So we actually start to accumulate data showing that, expressing this transposable element, okay, might create a condition for these tumors to better endure inflammation. Okay, let me take one step back and then say, so transposable element activity now is tied to immune response and inflammation. Okay, so the type of gene product that I've discussed today is new antigen protein product, right? And there are plenty of RNA product, including double-stranded RNA, right? So they are double-edged sword for two reasons. One is, yes, if you leverage them to activate more immunogenic events, that may be good to kill cancer cells. But cancer cells express them, right? There's an hypothesis and then some data supporting cancer cells express them to grow resistance to immune surveillance, right? They do that with a low level of inflammation. You actually exhaust the immune system, right? So cancer cells need to evade immune surveillance, right? So the tumors that we see, they all survived. Otherwise, we won't see them, right? So even though we're very excited to explore these things as a therapeutic angle, we definitely are very need to be very cautious about what's exactly happened, right? And that's not that can be that's actually a very exciting research angle to see whether or not transposable elements are actually conditioning tumor cells to be less sensitive to immune surveillance. Okay, I didn't go into this because I'm doing a little bit of marketing of the new antigen stuff, right?
Hi, Ting. Fantastic talk. I resonate with the last part that the epigenetic therapy will be the future. But I'm trying to understand, how would you have a cell type specific epigenetic rewriter? How do you know that only the cancer cell will be edited in the therapy?
Also a great question. You guys are really asking the fabulous questions, Short answer is we don't know, right? So at this point, the typical epigenetic therapy is global. Okay, Ting methyltransferase inhibitor, HDAC inhibitor, EZH2 inhibitor, all of these are chemicals that non-specifically targeted the entire genome. Okay, so there's very little specificity, both in the context of cell type and in the context of the genome. On the cell type part, a lot of them targeting cycling cells, so maybe we're all, somewhat okay with that. But the genome side, they're not targeting. Okay, how do we deal with this? So dosage, right? So I think, cancer genomes are already epigenetically destabilized. So it would take different amount of chemical to activate a particular target in a normal cell versus a cancer cell. That's the intuition, right? So we still need to collect a lot of data, build a better model to have precise control of the genome-wide effect of these non-specific epigenetic reagents, okay? The other part is site-specific activation, right? So far, everything is achieved through CRISPR system, okay? Now, CRISPR system is very powerful, so it certainly has the promise for the future. The biggest problem for us now is delivery, like, everybody's facing that, but I think many people are working on that. So ultimately, our hope is to be able to leverage the CRISPR system to do targeted site-specific kind of a manipulation, right? So we haven't done any of this. So you're absolutely pointing to the most important problems and also directions for us. But these are our current thinking. Okay.
Hi, thank you for the thought-provoking seminar. I think your answer to the previous question kind of already answered my question, but I still want to bring it out to hear your thoughts on this. So because we know TEs can have like a site-specific insertions, so they don't just randomly jump around the genome. And then recently there has been some discoveries on utilizing certain transposons, like a FONZAR or even L1 to do like a guided RNA insertion, which could be some like a therapeutic opportunity. So what is the current limitations in your opinion in terms of going down that road?
Okay, fantastic direction to go, right? But I want to make sure I make it clear that everything I presented today are fixed TEs. I haven't presented anything about the kind of the active, the newer, the polymorphic or even somatic T activity. If you want to know the preference of where T jump, don't ask me, ask John, right? Right? So that's a fascinating field. In fact, I work with many folks here in the somatic mosaicism across human tissue consortium to try to measure somatic transposon jumping activities across development across different tissue types. So there are definitely opportunities. There are also definitely causes for certain diseases. But what I have described, the focus today, are the TE activities that accumulated in our genome over hundreds of millions of years. They're fixed. They're no longer moving around, but that doesn't mean they don't have an impact on nearby gene, or they don't have the whole point is their impact is at the epigenetic level, okay? And guess what? I feel, that's probably an even bigger opportunity because the sheer amount of TE there in the genome, right? So the active ones are very interesting and important, but considerably way smaller a component of our genetic space, okay?
Hi, Dr. Wang. Thank you so much for the inspiring presentation. Actually, you already answered sort of my question. So I guess since you talk about a lot of the T's in the cancer genome, and I was just wondering your vision on the T's in the normal tissue development, and as well as you just mentioned the old T's, like the fixed one versus the active and jumping T's, which you would think that contribute more or potentially would be more important.
Right. I mean, depends on what kind of a time frame you're looking at, right? So if you think about this, looking at the fixed TE, you're looking at the kind of entire history of evolution, right? You're looking at the current active ones. That's important, knowing the mechanism and whatnot. But the evolutionary time is short, right? So you would see very different contribution if you just directly compare them and measure them, right? T is a mutagen, okay? It's a mutagen, no question, right? By being a mutagen, it also provides ability for us to evolve, right? There's nothing more than that, honestly, right? So what we see in our genome, either they have passed the selection pressure or they've been just drifting and then keeping around, right? So the new insertions that we encounter these days, I will have a very hard time to think that they be that important, like functionally, right? Most of them are neutral events. Most of them will probably go away in another million years if the species, we as a species, was still around, right? So it's just like a spectrum of evolutionary process. Okay. Yeah.
Thanks. Great talk. Of course, in cancer, a major problem is heterogeneity, sort of within the tumor, within cancer cells. From the data that you've looked at in your experience, do you have any sense of how truncal these epigenetic events are in terms of these aberrant transcripts or neoantigens? Is it present in a vast majority of tumor cells? Is it all the tumor cells, or is it only subpopulations?
Right. Also a great question, right? Yes, tumor, I was talking this today and I said we should never say cancer anymore. We should always say cancers because they're very, very heterogeneous, right? So two things. One is, we know they're heterogeneous, right? We actually have now evidence from, now you have to go through single cell, and for us, it has to be single cell long read, full-length RNA-seq to confirm this. So we have beautiful data to illustrate that. So we know, the distribution across a population, and as you expected, they're heterogeneous, okay? Therapeutically, it's not too bad a thing, right? So the idea with immunotherapy is not to make sure every cell responds, right? It's really to create a more healthy immune environment, right? So if you can, even if only 30% of the cells are expressing the right target and then you have a way to target them, you're actually going to create an environment so that the neighboring tumor cells get killed as well. So I wouldn't, I don't want the, what's the word? Perfection is the enemy of being good, right? So I think for this, it's good enough for us to pursue, right? But definitely not every single cell is doing the same thing. Okay.
Thanks.
Thank you so much for your time today and a wonderful talk today, Ting. So we have quite a few other questions to ask, but we run out of time. So maybe our trainee or our colleague can reach out to you later for their questions. But thank you very much for coming today, and let's join me to attend Ting's wonderful talk again.
Thank you. Thank you very much.